Everybody, welcome to yet another Journal Club brought to you by Life Extension Advocacy Foundation, LEAF. I'm Dr. Oliver Medvedik, and we're going to be doing another paper in our respective bunkers during our quarantine session here in the year 2020. So this is our July session. I'm here at my house in New York City. Everybody's in their respective homes, I believe, and we're going to be doing a very interesting paper. So I like papers that we've been doing some papers where things are starting to percolate into clinical trials, and I believe some of the work we're going to be covering here, although the work in this paper is not a clinical trial, it is there are clinical trials gearing up to implement this approach to reversing aspects of the aging process. And the paper that we're going to be discussing is from the Conboy lab. I'm going to share a screen right now. And okay, hopefully that looks pretty clear to everybody. So this paper was actually sent out, received May 13, 2020, which happens to be my birthday. And it's rejuvenation of 3 germ layer tissues by exchanging old blood plasma with saline albumin. And this is from Michael and Irina Conboy. And this is a paper by Medvedik et al. And they're over at Berkeley. And what this basically paper is discussing is a sort of a alternative to, so we've talked about approaches where you have a process called parabiosis, where you essentially you connect circulatory systems of 2 organisms, and usually to a young rodent and an old rodent, and very significant effects have been observed in many papers. You tend to see rejuvenation of tissues in the old mouse, and likewise, you tend to see some detrimental effects in the younger mouse. So it wasn't clear, it hasn't been clear for a long time what the effects were caused by. It could be youthful factors, so it could be factors in the young organism's blood that are, perhaps, replenishing what was missing in the old mouse, or it could be, a dilution of factors in the old mouse by the young plasma. And this paper here seems to suggest it's the latter, although it doesn't exclusively, there could also be additional young factors in the mouse's blood. But what's interesting in this paper here is they use something called apheresis, or what they refer to when they applied to mice, they referred to as neutral age blood exchange, or NBE. Basically, it's the amount of plasma that they remove from the. I'm going to stop sharing here for a second. The amount of plasma that they remove from the organism, they replace with saline, isotonic saline solution, and albumin, basically to mimic the osmotic potential of what they're replacing, and nothing else. So they're not adding any other factors. They're not adding any other blood factors, and they're adding this. They're replacing this plasma component in old mice, and also as a control in young mice. And I believe in this paper too, they also do a plasma exchange with the young plasma. So other approaches to this have been basically, a replacement of old plasma with young plasma, not just through parabiosis. Now, this technique here is very similar, this apheresis technique, where they do basically clear out somebody's plasma and then replace it with a saline solution that has albumin. The interesting thing about this is that it's already being used therapeutically in many indications. So if you just Google therapeutic apheresis, you'll find a wiki page, a Wikipedia article where there's a large table that tells you all of the indications that this is already clinically being used. So it's not being used, to reverse aging per se, but it is already being used clinically, at least in the United States, FDA approved for a number of different indications, and they kind of run across the board for, chronic conditions, such as if you have familial hypercholesterolemia, which means you have high levels of cholesterol. This could be used to clear that from the blood. Also, I believe I saw on the table severe cases of malaria. So I guess if you have a huge parasitic burden, that could be used potentially to clear the blood. But there's a huge table of things. So the cool thing about this procedure is that what I like about this is that it seems to be fairly simple. Most likely has a pretty good, I think, a good safety profile. It's been used already clinically for a long time. And if the effects are as robust or nearly as good as plasma exchange using young, youthful plasma into an older donor, well, this is less controversial. You're not using a blood product from another human. You basically, that's also extremely expensive. And I would assume that merely exchanging isotonic saline solution with a small percentage of serum albumin is a lot more cost effective and a lot safer. Let's get back to the paper where they actually do this in rodents. So I believe they do this in C57 black 6 mice. So they use a standard workhorse mouse. And let's take a look. And they look at basically the effects on 3 different tissues. So they're going to be looking at muscle, they're going to be looking at liver profile, and they're going to be looking at neurons in the hippocampus. So let's take a look. So just to get to the punchline, it's right there in the title. No secret, there is rejuvenation that's observed. So let's cut to the chase here. So let's scroll down. So those of you who really want to look at the statistical analysis of why they picked the sample size they did, it's in figure 1. They basically mentioned that the amount of mice that they used, is sufficient based on statistical criteria. And I believe because of this, they chose n equals 4. And then they used multiple samples, multiple tissue samples for each of their experiments. So basically, figure 1 just justifies why they chose that many mice, and not many papers really do that, go into that kind of justification statistically. So I thought that was pretty nice. But we're going to go right into the data here, which is where they look at basically the three different tissues, which come from 3 different germline tissues: mesoderm, ectoderm, endoderm, right, from developmental biology. And they look at muscle cells here. So rejuvenation of adult myogenesis and albumin-independent effects of TP. So what they call TP is therapeutic plasma exchange, I believe, is the term. And they have a regenerative index here. So images are on the left. So this is basically H&E staining, hematoxylin and eosin, which stains basically nuclei and proteins. So that gives you basically a cross-section of muscle. OO means basically old mice with old plasma, so that's their negative control. ONBE is basically old mice with this, where they do a 50% exchange with their, basically with their isotonic saline and albumin. And then you have MIHC hook staining, so nuclear staining, nuclear staining and staining of muscle cells. So you can see a larger percentage of muscle cells in the cross-sectional staining in left, and then you have a quantification of what they're looking at. So they're looking at regenerative index. So in these tables here, so fibrotic index is basically so regenerative index is basically, I believe, the amount of muscle cells. Fibrotic index is the amount of connective tissue or fibrosis, which goes up with old cells, and then minimal ferret diameter is basically the diameter of myofibrils, which is the muscle cells themselves. So you expect them to get larger, right, if the muscle mass is improved. And on these tables here, this on this index, these dashed lines, I believe, are comparisons to the experiments that they performed with actually young and old plasma, right? So all of the little dots here, your statistical spread here is the NBE, which is the, oh, sorry, it means, yeah, neutral blood exchange. So NBE, basically, that's their term for the isotonic saline with the albumin. So basically, fake plasma, let's call it that, right? So they're or artificial plasma, and these dashes are what you would expect in previous experiments with young and old when you have a plasma exchange. So right here, just by looking at this, you could see that with their sample size, you could see a pretty good correlation for regenerative index and fibrotic index, and also the diameter of the muscle tissues, the myofibrils, when they do, after they perform the NBE, which is the neutral blood exchange. And you can see that everything is shifted. So this YNBE is basically young mice that basically have received, an exchange with young mouse, or young mice that have received an exchange of their artificial plasma, and old mice that have received an exchange of artificial plasma. So they basically have taken out a similar volume of old plasma and replaced it with this artificial plasma, right? So you shouldn't really expect to see much of a difference for YNBE, so that's a positive control here. But you do see an effect with old where it's shifted towards basically the young parameters. And that spread is statistically significant, and that's what they saw with muscle tissue. So that was, that's quite surprising because, again, so these are samples from 4 mice, I believe, for, C57 black 6 mice, and looking at muscle tissue samples. And the fact that you're getting this effect after exchanging the plasma with something that's fairly neutral, albumin and isotonic saline, is already leading kind of, strong evidence that you're getting a dilution of some negative factors here rather than adding in, a positive factor. And again, this is only one tissue type they've looked at so far in this paper. They've looked at muscle tissue. So go back here. So they ask a number of different questions here, which is, okay, so you've got this response here. So they have another assay here. So this is, these are samples that they took directly from the mice. They have a proliferation assay where they basically validated it in a previous paper. So this is a proliferation assay for, I believe, muscle cells. Let me just double check. Figure 2 here. So which cell types did they use here? Dose response BRDU cells. I believe these are muscle precursor cells here. And what they're asking here is, well, is it the serum albumin itself? Is it the albumin that's actually causing, better proliferation of muscles? And at least for these cells, their answer is no. So this is basically, they have different percentages of human serum albumin. So this is actually human serum albumin that they're using. And by adding in larger and larger amounts, they basically, this BRDU assay, so bromodeoxyuridine is a kind of a classic assay that's used to test for DNA replication because it gets incorporated into dividing DNA. And they're not seeing any, growth effect here. So let me skip down over here to the bottom. What they look at down here is they're looking at, again, BRDU staining is in yellow, and hook staining is in basically, purple or blue, and that's basically just telling you all the cells that are, all the nuclei of each cell. And this is pre-TPE, so before they actually do the treatment. And these are basically cells from the animals themselves. So pre-TPE plus HSA. So after basically they do after they do the TPE, before they do the TPE, they add in human serum albumin, and this is post TPE. So that's the treatment therapeutic plasma exchange, and post therapeutic plasma exchange plus HSA. So basically, what this quantifies over here is essentially the exchange itself. Basically the dilution of factors, not just the addition of HSA or serum albumin, seems to be solely responsible for the proliferation of myocytes. So this is just a quantification of this. So pre-TP just means that these are sort of the negative control of the cell. This is how the percentage of cells that are dividing, and it goes up obviously after you do the treatment, but it doesn't go up significantly more if you add in human serum albumin. So we have a chat here. If this paper isn't a case for dilution or removal of cytokines from aged blood being more important than things in young blood, I don't know what is. Yeah, people with patentable artificial plasma mixtures won't be happy. Well, all is not lost here. I mean, this is an effect that's observed here. It might be better if there's additional factors. We're looking at only certain certain parameters here. They're looking at liver. I mean, mind you, they've looked at good representations of tissues from all three germline origins. It doesn't mean that there might be some additional beneficial effect here, but one thing that I'm going to kind of stop sharing here for a moment here, these apheresis or plasma exchange therapies have been going on for quite some time, and I'd be curious. I'd like to ask the Conboys if anybody has done basically epidemiological, tests out there to see what effects, if there's any kind of anecdotal effects of improvement. Actually, there are anecdotal. They mention, I think they mention at the end of the paper that that a certain percentage of, and I don't have the paper there. It's cited here, where there's a lower incidence of viral infections in people who, I believe, undergone this apheresis, but we'll get to that point in the paper. So let's, okay. So am I still sharing the screen there? Let me just close this for a second. Oh, I think I shut that off. Sorry about that. There we go. All right. So back to the paper. Okay. So it appears that myogenesis is affected. Now they start taking a look at the brain, because at least within the hippocampus, certain regions of the hippocampus, such as the dentate gyrus, I believe, cells still continue to divide. These were findings that that came about, kind of shocking findings back in, I believe, the early 2000s or late 1990s. And this goes down as organisms age. So they looked at a marker Ki67 for neurogenesis for dividing neurons, This is just a cross section from which regions? The subgranular zone SGZ of the dentate gyrus. So this is staining. I believe this is probably hook staining for nuclei. And the red is indicative of dividing cells. And this is just a qualitative image right here of, old mice, with old, basically just old plasma, and old mice with these neutral blood exchange happening. And you can see, you've got these little punctate foci here, Ki67 staining for cells that are essentially undergoing replication. And there's a quantification here where we basically have, the number of cells per SGZ, in the dentate gyrus, young, so young mice that have received young plasma, young mice that have received the neutral blood exchange. Seems to be a bigger spread here, actually a bit higher, which is interesting. I don't know if that's an artifact or if that's real, but it's it's certainly not significantly lower. And then old that have a neutral blood exchange, and then old, which is doing pretty low here. So so right now just by having a neutral blood exchange with serum albumin and isotonic saline, you do seem to improve, the ability of regions of the, hippocampus to basically divide. So this is just a control panel for the antibody staining that they're using, to make sure it's not cross-reacting. So that is really interesting. So you're getting an effect both in muscle tissue and neurons. But there is something interesting that they mention here. In the muscle tissue, I believe this is in supplemental, it's not here. In the muscle tissue, the effects seem to be, independent of concentration of serum albumin. I believe that there was an effect here with higher dosages of serum albumin. I have to find where they mention that. Let's see, this is figure 3. For the life of me, I don't know where I caught that paragraph here. Probably further up here. So no effect with serum albumin, but anyway, I'll find that later. But they do mention in the paper that as we age, your blood-brain barrier gets leaky, and it was noted that that other studies have shown that having higher levels of serum albumin tend to be not very good. It promotes inflammation, and if it's correct that you get improved hippocampal division with more serum albumin in this paper, my question is that because the serum albumin here is better? Is it not misfolded? Are they using fresher albumin? And is it just as you get older, clearly proteins misfold as you get older? Is it is it the quality of the serum albumin? I'm not sure. I'm not sure if that was exactly addressed in the paper, but that's a question I would ask the Conboys. I'd be interested in that. Okay. So let me go back to the paper. Let's see. Any questions here? Nope. Still patentable artificial plasma. Yeah, there might be still useful factors in young blood. Okay. So on to the liver, and no big surprise, you do get beneficial effects in the liver. So I believe what they look at in the liver for for positive or detrimental effects is adiposity index, meaning the liver accumulates kind of fatty tissue. So that's a bad thing. Or fibrotic index, meaning the liver accumulates connective tissue, which, to the detriment of the cells. So for a healthy liver, you want to have a lot of healthy hepatocytes so they can perform their function, and you don't want any of the other stuff accumulating. And they use oil red staining for fatty deposits, and they use this, I believe, this Masson's trichome staining for connective tissue. So blue is connective tissue. So oil red, you've got these little deposits here, which this is old, so old plasma in old mice, and this is ONBE, so old with nuclear blood exchange. So basically the isotonic exchange with saline and plasma, saline, and serum albumin. And both of these indices improve, meaning that the amount of connective tissue goes down and the amount of fatty deposits go down. And that's quantified here in this adiposity index, where young is basically your positive control with young mice with young plasma. And then YNBE is your, your artificial plasma. And then the one that we're interested in is the ONBE, right? That's the old mice having the neutral blood exchange. And you could see that it's relative to the old mice with the old plasma, it's shifted downwards considerably. Those are nice. It's nice to see quantifiable data where, yes, you got the error bars, you have the statistics, but you can just see it with your own eyes that, you get a pretty dramatic effect. You don't really need to, do any kind of really deep statistics to show that this is having an effect. Let's see. We have something in the comment section. I'm going to stop sharing this. Yeah, if anyone has a comment here, that would be good. So we've gone through basically the bulk of the data here on these mice. And the next kind of image is basically going to be doing a proteomics analysis. I won't go into in a lot of detail, just to show, the proteomic analysis essentially shows that a lot of the protein expression profiles get shifted towards a kind of a younger mouse. And there's a lot of, you could dig through that proteomic data and see if there's your favorite protein goes up or down. We could take a look at some of examples in there. But are there any comments coming in? Again, this is just an exchange with a isotonic saline solution with albumin that's supplemented. There's nothing really else, and the volume, I believe, is 50%. So I'm not sure. So for human therapeutic purposes, I think they have two different types of. I think it depends on what procedure you're undergoing. And it depends on how much of your blood plasma gets exchanged. Essentially, my crude understanding is that your blood is basically pulled out, centrifuged. So all of your basically red blood cells, white blood cells are pulled out. The plasma fraction is pulled off and then replaced with, an isotonic saline with albumin to maintain basically, the right osmotic index. And then that's put back into your body, right? And that procedure has been around and it's been patented since the 1970s. So that's and again, if you Google plasmapheresis or I think anapheresis, I might be mispronouncing that. Those terms are used interchangeably. And it depends. I think the anapheresis is kind of a much larger volume that's exchanged in humans. Plasmapheresis, it's less of a volume. Yeah, so I don't know how many people have undergone this, but probably, I would say, if not millions, then many, many thousands, right? So, okay. So let's go back to sharing screen. Okay, so I won't go through this in much detail. So this is basically comparative NBE and TP proteomics. So, so therapeutic plasma exchange and their neutral blood exchange. They compare the two approaches. So it is a bit of a different profile. Let's take a look here. So old versus young mouse. So old mice and young mice. So I believe this is basically this is different protein expression profiles. And this is, you could see that it's not, it's not 100% replicated, right? So this is a pattern of different proteins. So they're not marked here. So kind of, I can't really go into any detail here, but just as a snapshot view, you could see that the profile is roughly similar in that you have the shifting of certain proteins in the same areas as you have, old versus young mouse versus an old neutral blood exchange affected mouse. So there could be additional, factors that are at play that haven't been introduced, in this system here for the the neutral blood exchange that could still play an effect. These are factors that basically change on the right, so levels go up. So this is, if I zoom in here, so after nuclear blood exchange, certain things, are kind of go up, and elastin, for example, or maybe that's not elastin, no. Erythropoietin, right? So here's factors that improve, that cause differentiation of blood cells to erythrocytes. So basically, factors that you are expect to see go up to promote basically differentiation of cells. A lot of these factors go up, growth factors. So no doubt there's probably many more. So I'm not going to go through, so they're all categorized here by angiogenic factors, tissue homeostasis, and so on and so forth, right? So these are all affected in this procedure here. So, it's a pilot study, and they mention here that this is actually going into, I believe, phase 2 and 3 trials. Scrolling here. This is what I wanted to mention. So it's down here. Promising work in progress follows an interesting observation that TPE, and that's when they do it in humans, therapeutic protein exchange, plasma exchange, anecdotally reduced incidence of viral diseases to 0 in patients over the course of a year, when for just flu-related hospitalizations, 60% were from the same age group. That's pretty dramatic. I don't know what they mean by anecdotally. So this was, something probably that medical doctors notice. I'm just surprised that in the medical establishment, better records aren't kept of these types of observations. This is this type of, but in all fairness, nobody expected to see any kind of age reversal effects, using this type of application, using, what they, when it's done in humans, referred to as, I believe, therapeutic plasma exchange. And in this model here, NB, neutral blood exchange. So now that this paper's come out, and now that this, these observations have been, quantified in mice, and the Conboys are going ahead with kind of more rigorous clinical trials. I'm just thinking out loud here. I'm just wondering if maybe, this is a question I'd love to pose to them, if they're not working on a protocol to just give to medical doctors that are, doing these types of procedures already, right? If there's a way to basically capture the data that's out there already, because if anecdotally it's already been noticed that incidences of viral infections, particularly the flu, have dramatically decreased in patients that are elderly undergoing this therapy, clearly, viral infections are foremost on everybody's mind now because of pandemic. But there's a lot of data there that can be captured and basically looked at through a statistical lens. And I'm just wondering if if there's a protocol in place, or if the FDA has, or not just the FDA, or for any other, any other medical or drug agency around the world has in place to kind of plug into an already ongoing series of therapies out there, right? So somebody just mentioned here, the albumin is only used to maintain the density of blood after protein density goes down. No other effect. Yeah, albumin is basically, it might have a bunch of other effects, but classically, when people talk about albumin protein, it's there to maintain the osmolarity of the blood. So basically, as your blood circulates around and around your body, there's liquids, right? H2O, all the other things that certain small molecules diffuse out of capillaries, and then things diffuse back in, right? So and that's under pressure, and albumin is there to maintain the proper osmolarity, so you don't get edemic, and liquids don't pool around your body. So it's basically it's super useful there to have proper exchange of factors taking place across capillaries throughout your body. There could be some other roles there as well, but maintaining the proper osmolarity of blood, I believe, is one of its key effects. Amanda asks, how long would these rejuvenating effects last? I don't know. That's a good question. Do you only see a boost in neurogenesis shortly after the exchange, and then it tapers off with time? A quick scan shows little indication of general health benefits of plasmapheresis. Whether that's because they're not noticeable or don't exist, I don't know. Yeah, again, that's why a better study is warranted. When people do plasmapheresis, I don't know if doctors are noticing this or people are taking notes, right? What is the subtle effect, right? If somebody's health profile or somebody's aging reversal is 5% or 10%, that's a small number. But if you kind of spread that across an entire population, that's massive, right? I mean, who wouldn't? I'm just tossing out numbers here that that may not reflect reality. But if you lift 10%, if your aging was reversed, or your rate of aging was slowed by 10%, would you notice? I'm not sure. But you would have to do a before and after, right? So, whether, would you notice if somebody lived to 100 versus 90, right? I mean, a lot of these things, if you don't really, capture them, if you don't look at them statistically, nobody really notices, right? If you don't really keep an eye on these things, and if the parameters you're looking at for plasmapheresis are just basically focused solely on the therapy itself, meaning that your cholesterol levels go down, then why would anybody look at anything else, right? And if somebody's cholesterol levels go down, maybe they're more active and maybe they feel better just because their cholesterol levels go down, and whoever's administering the therapy might just solely attribute it to that, right? I mean, nobody's doing punch biopsies to see if their muscle mass is improved, I think, as a consequence, doing this type of plasmapheresis. So the question is are they really, this is a lot of lost data potentially. How long will last? Why was there no test of lifespan? Well, probably because they wanted to publish this paper within a relatively short period of time, and basically promote the stimulating conversations that we're having right now, and get us all to say, hey, let's do a lifespan assay next, right? Which they succeeded. So I'm all for publishing data as soon as you have enough data. Obviously, you can do everything in the kitchen sink and have a paper that's that's been kind of ongoing for a number of years, and that's fantastic too. But there's something to be said to when you basically have preliminary data that's statistically relevant to just get it out there and say, hey, this is a direction we should all basically um, take a look at much more rigorously, which is what they did here. So that's kind of my 2 cents on that. So
Well, we know, Oliver, just to interject, we do know somewhat why they didn't do a lifespan study for the Conboys, because quite honestly, that's not the way that they roll. They've never done. Their objective here is to demonstrate rejuvenation and boosting of stem cell activity. That's what they're after. They're not necessarily at this point concerned with, oh, does it make you live longer? They're more concerned with the tangible, immediate effects. That's what they're trying to establish, and that's the way that they're going, so they can get it to human trials sooner. I understand why you'd probably want to do a lifespan study, but they're incredibly expensive, and it's just not the way they're going with it. It's not the style that the Conboys use.
Yeah, and one way to look at it is it's generous of them to publish this here, because any labs that have already geared up to do lifespan analyses now they've got it's easier for labs now to get grant funding to go ahead with these types of lifespan studies using neutral blood exchange, because now they have the Conboy paper to basically cite and say, look, this suggests very strongly that we're going to get a lifespan effect, so give us the money to do this assay.
Well, rejuvenation, I'm gonna argue that rejuvenation of tissues, or regeneration is essentially, rejuvenation is reversing something from an age state to a more youthful state, in which case what the Conboys, and going back to 2005 with their work, they've already demonstrated that that's possible. So rejuvenation does occur. I understand what Didier is saying, that rejuvenation in the context of lifespan, but that's not what the concern is here. But it is definitely rejuvenation.
Yeah.
Depending on your definition.
Yeah, so definitely, good follow-up is looking at, looking at other aging biomarkers such as methylation status, and of course, the gold standard, which is lifespan extension. And no doubt we're going to see follow-up papers on this. But that being said, this certainly is strong evidence that we will see a positive effect here. And I think they have enough data, and they certainly do have enough data to go ahead with clinical trials for the two reasons that I mentioned. One is that the data in this paper, and number two, this type of approach, this therapeutic plasma exchange, has been going on for decades. Since I've been born, practically. So it's a procedure that's already taking place, and why not take advantage of that and basically try to capture additional data, right? So so that's what I kind of love about this, is that we have a procedure, it's ongoing for a whole list of different indications, and if you can just pick the right indications out of that list and pick the right groups of people, that are having this therapy anyway, young versus old, you pretty much have, a clinical trial that's kind of already there. It's just the data is just not being looked at properly. So I'm really psyched about that, is that we potentially already have, a massive, therapeutic plasma exchange experiment taking place out there. It's just not being looked at from the aging perspective, right? And of course, why would it be looked at from the aging perspective? Because the medical establishment doesn't recognize aging as a disease, so they would never look at the aging perspective. You would look at a whole bunch of other indications. But again, that can also be looked at. That could also be, I don't want to say jerry-rigged, but that could also be set up in the right way, where you look at multiple modalities that are indicative of aging, right? And look at them and look at different profiles that correlate with, biological aging, physiologic parameters, and see if these physiologic parameters improve. And so if you look at physiologic parameters like, like gait and reflex response, and maybe tracking response with eyes to, to different types of, stimuli on a screen. And then you also look at methylation status in these patients that are already undergoing these. There could be some really promising data there that's not being captured. Reason think the reverted state persists given the many feedback loops in the system. Yeah, I don't know. I don't know how quickly it would attenuate itself. The question is, would this effect need to persist if, would additional factors be required? Is this effect only temporary, having this type of plasma exchange? I think there's a model in this paper, which I didn't go into, which they think it might, well, actually, they don't go so far as to kind of suggest that it would persist. They have a model there where it shows that how these feedback loops might be reset. But how long that would last, I'm not sure.
Quite some time, I think, Oliver. Bearing in mind that I've spoken at quite a lot at length with Irina and Michael in recent months. I interviewed them last month, and I spoke to them again last week. They do believe that there are a lot of feedback loops built into the system, so that there is quite strong reason to believe that the state may be persistent for some time. Obviously, there's no firm how long figures on that, but I mean, what they're saying does make sense if the younger milieu, if filtering it actually effectively almost reprograms cells and tissues, it could be quite a persistent state for some time. I'm thinking in particular in the context of the stem cell niche, which is where all the magic happens. If you expose that to young milieu, then you could have quite a persistent state. And just to circle back to a question we had earlier, do we know what some of these factors are? I'm going to say that based on some of the earlier work from the Conboys, I think you're going to find TGF beta is definitely in there, TNF alpha. You're going to find things like notch signaling is important. What's the other one? AMPK, MAP kinase. That's another one that they've looked at. They reckon, Oliver, that there's only probably maybe a handful of master sort of controllers that sit atop this whole pyramid of cytokines, and if you adjust them and attenuate them to the not block them completely, because obviously your body needs them, but actually attenuate them to the point where they're more youthful, it should then have a cascade effect and affect all the hundreds of other molecules underneath. So but it's quite exciting.
Yeah, and the fact that we have neurogenesis happening at an elevated level in the dentate gyrus here, it does give evidence that it is a lasting effect, because you're not going to get that effect without some nuclear reprogramming taking place. And if again, and I'm no doubt people are going to look at the epigenetic status and the changes that occur. If they do occur, then again, that's more evidence that this is a lasting effect if you get an epigenome being basically reset, right? So of course, things are complicated in real life. How much of an effect you get, you still have other senescent, in an old mouse, right? You still have other senescent cells that are lingering around that might be pushing the whole system back into, in the other direction again. You might have this tug of war. So
I'm going to just interject in there and just say and remind you about the Katcher paper, Harold Katcher, when they did their elixir thing. They're saying that their level of senescent cell SASP, which would be indicative of the presence of senescent cells, was reduced as well. And I believe the Conboys may have even looked at that as well. So there is some possibility that in resetting the age of the blood, the blood, you may also be somewhat addressing senescent cells, because of course reducing the inflammation allows the immune system then to start working better and then start clearing house. So there's an argument to be made there as well.
Mm-hmm.
I think it's quite exciting. It could have a very broad, potentially a very broad, wide ranging effect.
Yeah,
it could do.
Yeah, I mean, again, I'd have to take a look at the papers where they actually do this in human therapies, because there is one paper that was cited here. There might be, there, just to make sure what any potential negative health consequences are to dilution, because I think there are some. I don't really know some of them. There are side effects where people experience tingling and things like that, and because you are basically taking out a substantial amount of blood plasma and replacing it, and then and then components that are in that blood plasma have to basically be resynthesized by the body, right? So it's yet to be seen, but we do have the infrastructure to do to do these do these experiments. They're already being carried out therapeutically, and it's a lot safer than doing a human to human blood plasma transfusion. And the amounts that are needed, in parabiosis experiments, if you're talking about 50% volume, I mean that's a lot of volume, and that's a
It's just not practical, Oliver. I mean, at that kind of scale, it's just not practical to use blood from other people. Michael Conboy used the analogy. It's a little. I use an analogy that was like an oil change. He said it's like a fish tank changing the water. And I think it kind of works. It makes sense. It's kind of like with a car, you go in for an oil replacement. Well, that's what you're effectively doing here. You're taking your own blood, you're taking it out, you're scrubbing it clean, and you're returning it. That logistically, from a logistical point of view only, is far more practical than having gigantic vats of young blood being produced, or blood. You've probably seen the meme, the blood boy, the blood boy. Like rich millionaire. It was doing the when this all started a few years ago.
Oh, with what's his name, the billionaire? Okay.
Yeah, who should I'm not naming him because we but yeah, you know who. But it was all to do with the parabiosis, and everyone was talking about Countess Bathory and Dracula and Blood Boys, and it was like these rich billionaires were going to have a number of young people to take blood from it. It's obviously very silly, the idea,
right?
But this circumvents it completely. Why would you need blood from anybody else when you can use your own? It just needs recalibrating.
Yeah, it's interesting. This kind of reminds me a little bit about the controversies still swirling around about embryonic stem cells, right? And then things start to shift to induced pluripotent stem cells, and a lot of the controversy kind of went away. Although it's still not clear if induced pluripotent stem cells recapitulate everything in embryonic stem cells, but it kind of reminds me that, right? Because this is a zero controversy type of controversial approach. Like you use the analogy for an oil change there. I mean, people understand that. That's nothing controversial about that. And we are talking about isotonic saline with serum albumin, which I don't know where they get the serum albumin from. It might be recombinant, but it's a simple protein. You can derive that from a number of different sources,
including bovine. You could use bovine, which is very common.
So and again, what I also love about this is that it's already being used in a number of different interventions therapeutically. So all the FDA hurdles are pretty much gone. It just needs to be, I think, very intelligently applied to the right cohort of people. Now, you can't say you can't set up these trials and say we want to cure aging, but you can certainly set up trials, and I think it's kind of similar to the TAME trial with Nir Barzilai. You can certainly, I mean, he's having some difficulty, I think, setting that up, but it's certainly, worth setting up trials where you look at several modalities that are, or several indications that are as a consequence of aging, right? And basically see if all of them, essentially all of these parameters improve. And each of these different indications in of itself as a standalone can be a disease, right? Such as perhaps liver fibrosis, things of that nature, right? Maybe certain dementias, so on and so forth. Or initially you might want to look at less complicated things that are much more kind of easier to quantify. But it's certainly worth looking into and kind of aggressively setting up clinical trials, because I think the infrastructure is already there, and we may as well go ahead with this. So either it works or it doesn't, but the infrastructure to find that out is already in place, is what I'm saying.
Well, yeah, obviously I can't say much right now because I'll get told off, but we are in discussion with the Conboy lab, potentially regards human trials. So so keep watching this space. We may have something to announce officially soon, but we are looking at the plausibility of maybe doing a community initiative which helps get the human trials off the ground. I mean, obviously, it's an extremely expensive endeavor, but there are things in motion, and we're very interested in that. So, yeah, watch this space. Hopefully, we'll have some more news in the next few months in regard to that. But I'm tentatively excited. But I certainly think we're going to definitely see human trials for this approach, maybe this year.
Yeah.
Maybe, we're talking imminent because they're ready. They're literally saying, look, we're ready to do it.
Yeah.
So let's do it.
Yeah. Yeah. This reminds me a lot. This is to me like sort of a, this reminds me a lot of metformin, right? It's a procedure. It's a drug that's been around for many decades. It seems to have an excellent safety profile. It's being used for other indications. And here we have, like with metformin, when you had epidemiological data suggesting that people live longer on metformin, humans that are diabetic versus non-diabetic humans that are not taking metformin, right? So and we've got all these animal studies suggesting that metformin targets basically, pathways that are responsible for, promoting longevity. And it seems that we have a lot of data here suggesting that we have this intervention that's a little more invasive than metformin, but also been around for a long time. So therefore has established protocols and procedures, has a good safety profile, has medical practitioners that are already used to delivering this type of therapeutic, and lo and behold, there's data now suggesting that it may have also additional effects that are targeting the root causes of aging. So that's great. We can have another session where we talk about very low hanging fruit interventions that should be tested, should go into trials immediately, like metformin, for example, and see if maybe these in combination would even be better. I mean, metformin and therapeutic plasma exchange, would that have an even better effect? I don't know. I'm super excited about this for all these reasons. Just because it's I would be surprised if we wouldn't get any beneficial effects, because all the evidence that we have points to there being beneficial effects.
Don't forget all the anecdotal accounts as well. And yes, I know it's anecdotal only, but a lot of people in in the past have reported from just simple blood transfusions, like giving blood. They've reported that they feel, better or healthier if they do it regularly, which again, it's plausible because you could be actually reducing the amount of cytokines and things in your bloodstream that are affecting, your health. So I definitely think there's going to be some movement on this soon. Hopefully lifespan can be part of that, and therefore everybody here can also have the opportunity to get involved. Didier has mentioned mouse lifespan studies. Not so practical in humans, unfortunately, Oliver. So I guess what they would do in human trials in that context, they'd maybe follow the patients for a year or two and then look at various aging biomarkers and see if they're changed. It is unfortunate. I'm going to say this. I want to say this earlier, that the Conboys are not really keen on methylation clocks and epigenetic clocks of various types. They're not really big fans of that. They prefer sort of the approach of looking at cytokine levels and comparing them. They don't tend to sort of go in for the methylation clocks. I would like to see that those biomarkers used as well, to be honest. I mean, yeah, I get where they're coming from. They're saying, well, look, this is tissue regeneration. It's demonstrable. It's happening right now. There's the proof. But I'd love to see what's happening on an epigenetic level as well.
Yeah.
So,
well, there's a lot of other labs out there that can take this and certainly run with it. And again, I'd like, unfortunately, a lot of health information is siloed here. It would be fantastic if researchers could find out, what's taking place as far as these therapeutic plasma exchanges are occurring across the country. Maybe it can't be done in the US easily. Maybe it can be done in Japan or some other country where they do this, where you can have a list of, I want to set up a clinical trial where I take a look at 1,000 people that have this health profile that are between the ages of 60 and 80. And I want to also take a look at people undergoing this same the same procedure that are between the ages of 20 and 40. And that's going to be my young cohort. That's going to be my old cohort. And basically, run your therapies the way you normally run them. And I just basically want to have blood samples before and after from these individuals, and then we'll analyze them. But like I said, it's kind of a shame that there's nothing in the infrastructure to be able to take advantage of stuff that's already ongoing. Like with the metformin study, where they basically saw an improved lifespan in people that have been taking it. That just happened to come after the fact of having all of this epidemiological data that was unlocked, and researchers were able to look at it and say, whoa, we've just noticed something here that's pretty kind of mind-blowing. And that data was unlocked, and people were able to basically access the data and find something remarkable. So it'd be great if we had a system in place that can do that with this one approach as an example, this one intervention or this therapy as an example. But there might be other therapies out there. There might be some generic system that can be put into place where this data is not lost. But maybe I'm asking for too much out of the healthcare system here in the U.S. Maybe a country like Estonia can pull it off, where everything is basically on electronic files, and they're able to do this much more easily. I don't know.
I'm sure we'll get there anyway. So so we'll see if we can help out in any way to push the needle along. So, and more on that later, maybe. Yeah, it seems to be the low-hanging fruit. We talk about low-hanging fruit a lot, but it is one of the low-hanging fruit, isn't it?
Yeah.
Fatima's laughing now because she hears low-hanging fruit all the time. It's literally our buzzword, yeah. but it is an example of low hanging fruit. It's technically, I mean, you could do it right now. There's absolutely no barrier to doing it. You could go and do it now if you wanted to. The technology is available.
Yeah.
It's just a matter of time now.
No barrier except time, money, and motivation. But certainly no technological barrier.
Yeah, I mean, in principle, you could go and do it this afternoon if you wanted to. It's absolutely. I mean, I'm not going to say it's simple, but it kind of is simple. It's kind of a low-tech approach.
Yeah.
Which is why I like it so much. And it's also possibly why the results that we see in animals are much more likely to translate to humans as well. Because the approach is targeting something that's pretty fundamental.
Yeah.
So exciting times.
Out with the bad, in with the good.
Well, that's literally it, isn't it? It's literally it. I'm still on the fence about the whole, stuff in young blood, because honestly, I think there is still stuff, potentially useful things in young blood to be found. So I don't think things like alkahest are necessarily barking up the wrong tree. I think there could be some benefits from that.
And before I lose this train of thought, there could also be, you might also improve the turnover of misfolded proteins by this activity, and one thing I'd be interested to see is as a consequence of this, do you remove are there proteins that are oxidized or misfolded that basically are cleared out, and do you have an improved ability, would this somehow prime the cells to basically degrade misfolded proteins and synthesize ones that are more functional, right? Rather than just a mere depletion of well, I mean, this might suggest it, right? Because you're depleting factors, right? So if you're depleting it might just not be a certain negative proteins going up, but it could be just also clearing out misfolded proteins. But that in of itself would be another experiment to basically take a look at what's going on there.
So yes, Paul has said a barroom discussion suggests that the treatment might be available right now at a price, although he's got no evidence of it. Well, this is also another way, Oliver, that things can happen sooner. And I realize it's a controversial or potentially controversial area of medical ethics, but paid patient participation in human trials, where someone wants to be part of a trial and guarantee that they get this new cutting edge treatment and they're willing to pay for it. Is it wrong? Is it right? My personal opinion is I don't see an issue in it. I don't think it should be an automatic taboo.
Yeah, I think if if people are adults and know what they're signing, and the person that's carrying out these procedures is a licensed medical practitioner, and the data is properly collected, then I mean, it's your money, it's your time, your will, right? You're not forcing anybody to do anything. So as long as things are on the up and up, and it's not some shady clinic doing things in a non-sterile fashion, then as long as the means are done properly, right? As long as it's carried out in a proper fashion, then sure, why not?
Informed consent at the end of the day. That is the way ahead. And let's face it, if that's what it takes to push the needle, there are certainly going to be people who have the means, who want to pay to participate, and of course, in doing so, that will dramatically reduce the the overheads that researchers need to find in order to do these. So, I think as long as it's done correctly, I don't really see it being impossible to do. So, fingers crossed we can get this into human trials and done, and who knows?
Yeah, and who knows? There might be there's a lot of indications for these therapies, and if the anecdotal evidence that viral infection rates decrease dramatically in the elderly as a result of this, the current pandemic may have an unexpected benefit to pushing this forward dramatically for at least that indication, and as a side consequence, we get to see whether or not it affects the aging process as well, which most likely it probably will if we think it's working the way we think it's working.
So there you go. And speaking of that, I did hear that some people in, I think, Russia, a group of researchers are actually using that technique as a potential therapy for COVID. So who knows? It may find application and support through that as well from the wider medical community. Who knows? But it's exciting. I'm personally very excited about it. And I can't wait till we get to crunch time and start testing it, because I think there's enough evidence there now, and the FDA isn't going to have an issue with it. So let's do this.
Yeah. Okay. A little bit of good news coming to you from the year 2020, which is much needed.
So anyway, it also makes me it also makes me wonder what Irina's going to be speaking about at the conference, because of course she is going to be one of our speakers this year, and that's really not very far away now, is it? So yes, good grief, another month. I'll tell you, another month has just disappeared. Where does this time go? But we're going to be holding a conference on August the 20th to 21st. It's a virtual one, so everybody can join in potentially, and Irina will be there presenting some research, so maybe she will have some additional news that we didn't know.
So weird. For some reason, I still think it's April.
Well, that's how it is with lockdown. It's like, oh yeah, what day is it today? And I'm like, I don't know. Does it really matter? Yep. I don't know if anybody else has got to that point where you just go, well, I don't know. Is it Monday? Oh no, it's Friday. Oh, yeah, whatever. We live in some strange times, you see.
Yep. Okay, everybody. Well, I think we've covered the salient points of the paper. We've, I think, had a good virtual roundtable discussion here on the past, present, and future applications of this technology and other low-hanging fruit technologies, so to speak. And yeah, I hope to see all of this, quickly tested and if positive effects come about, swiftly implemented. And if neither of those two happen, I will be sorely disappointed.
There you go. You go, people. Get on with it. Otherwise, Oliver's going to be sorely disappointed. So everybody, talking to you, FDA and everybody else, you've been warned. So, all right. on that silly comical note, I'd like to thank everybody who's joined us today, obviously via the stream on Facebook, and heroes who are here with us on the call today. They've all been a bit shy, but there has been a little bit of chat in the text. So great to have everyone here, and thanks for your support, and all the other heroes who are not here today with us, but who do support us every month. And that really is a massive boost and helps us to keep on doing things like this and all the other wonderful things that we do. So thanks for that, everybody. And if you're interested in becoming a Lifespan hero, check out /heroes, and also check out the conference as well. So more details on their website at lifespan.io, and you can't miss it. You literally can't miss it. There's a banner at the top. So come along and join us for that as well if you can. And I guess we'll see you next time, and we'll find something else to talk about. I'm sure we won't be short of the topics.
We're never short of topics.
We're never short of topics. So much going on. All right, guys and girls, everybody out there, stay safe, and we will see you next time.
All right. Take care.